Hello everybody, my name is Leah Weber and I'm a graduate student at the University of Illinois at Urbana-Champaign. And on behalf of my co-authors, Palash Sasito and Professor Mohammed El-Kabir, I'll be presenting our work on a method of doublet detection in single-cell DNA sequencing data. So as many of you are aware, cancer arises as a result of an evolutionary process. So very briefly, a single founder cell gains a somatic mutation, and over time this cell divides and may gain additional advantageous mutations, giving rise to a clonal expansion or the growth of a tumor, with the end result of this process being a heterogeneous tumor made up of a mixture of distinct clones. And single-cell DNA sequencing has been a particularly exciting advancement because it provides a new data source to explore and quantify the heterogeneity of a tumor, as well as allows us to reconstruct its evolutionary history with high fidelity. And ultimately, these analyses can help us get closer to our long-term goal of optimizing cancer treatments and gaining new insights into the evolutionary patterns across patients. So the term single-cell DNA sequencing is really an umbrella term that covers a wide array of experimental techniques. So our work is focused on amplification-based methods that are ideal for identifying point mutations and typically have medium to high sequencing coverage, such as MDA. So I'll just briefly summarize this pipeline for anyone unfamiliar with it. So first, a single cell has to be isolated from a sample, typically with well-based protocols or microfluidic droplets. So throughout the remainder of this talk, I'll use the term droplet to refer to the outcome of this isolation process. Then the DNA must be freed from the cell nucleus, and a unique identifier is inserted into the droplet so that we can associate downstream analyses with a particular cell. After that, the DNA has to be amplified to increase the amount of material for sequencing. Then the fragments are read so that point mutations can be identified. And even though there are numerous advantages of working with single-cell data, our researchers must also contend with a number of drawbacks as well. And a number of these challenges are associated with the amplification stage. So you may have heard of allelic dropout, which is a very common challenge. And this is where an allele fails to be amplified or is said to drop out. And this means that if there's a mutation present in that cell at that position, we won't observe it in our sequencing data. Biases towards certain alleles can also occur, and also confidence stakes that occur during the amplification chemistry can lead to false negative and false positive sequencing reads. In addition, errors can also occur during the isolation stage, where instead of isolating a single cell as intended, two or more cells are captured in a droplet and linked to a single barcode. So this is what we refer to as a doublet and is the focus of our talk today. So you might ask, well, cells are pretty small, does it really matter if we capture two instead of one? And the answer is yes, sometimes it can. So in fact, we can further break doublets down into three categories. So the first is a term that we borrowed from the single-cell RNA sequencing literature, and it's called a neotypic doublet. So a neotypic doublet occurs when the mutations present in the doublet cells form a novel genotype that's actually not present in the tumor. So this can make it more challenging when we go to try to infer the clones of the tumor, as well as decipher the mutation co-occurrence patterns. In addition, we also have nested doublets, where the set of mutations in one of the cells is a proper subset of the other cell. Or we can also have a cell flip, where the sets of mutations in the two cells are identical. And although these are less problematic to the inference problem than neotypic doublets, they can also cause errors when we go to estimate the clonal abundances. So what are the current options that exist for dealing with doublets in practice? Well, this problem has been pretty well studied in the single-cell RNA sequencing literature and typically involves machine learning to identify likely doublets prior to downstream analyses. However, the disadvantage of applying these methods directly to DNA sequencing data is that it's not optimized for nested and self-loop doublets, and it also doesn't account for the unique error profile of single-cell DNA sequencing. So on the DNA side, the main approach has been to account for doublets as part of the inference process. So this has helped minimize the impacts of downstream errors, but typically significantly increases the complexity of the inference problem. It also doesn't do a great job at identifying the nested or self-linked doublets. So critically, we noted that there was no standalone methods for doublet detection in single-cell DNA sequencing data. So this is what motivated us to develop doubletD, which is the first standalone method for doublet detection in single-cell DNA sequencing. So the key insight of our method is that the frequency spectra of the observed variant allele frequency, or VAF, which is the ratio of variant reads to total reads, will be distinct for doublet droplets and singlet droplets. And so given these observed read counts, we can actually tease this out using probabilistic inference and our understanding of the single-cell DNA sequencing pipeline. So the question that doubletD answers is, given observed total and variant read counts, is a droplet a singlet or a doublet? So to do this, we use a probabilistic graphical model to describe the generative process for the observed data. And while I won't have a chance today to discuss it in detail, I want to take a few minutes and describe the model at a high level. So our generative model consists of 4 submodels. We have the doublet model, which models the doublet status, the genotype model, which models the variant allele frequency in the droplet. Then we model the common challenge of allelic dropout and the latent VAF that arises as a result of that process, followed by the generation of sequencing reads in the read count model. And I'll just take a moment here to go into these 4 submodels in a little bit more detail. So first, in the doublet model, we model the doublet status ZI using a Bernoulli random variable with success probability delta, where delta is the prior doublet probability. And if delta is not known, we can actually do a sweep over possible values and then just take the maximum likelihood solutions. So in the genotype model, we model the variant allele frequency, or the VAF, which is the ratio of variant to total alleles in a droplet. And the value that the VAF Xij can take is actually going to depend on the doublet status Zi. So when Zi equals 0, meaning it's a singlet, there are 2 alleles at each locus. And the VAF Xij could take on the values of either 0, 1/2, or 1. But when Zi equals 1, meaning the droplet is a doublet, then we have 4 alleles at each locus. And the VAF Xij can actually take on a greater range of values, including the value of a 1/4 or a 3/4, which we don't see with a singlet. And here I want to point out that we're making an assumption that each locus is copy neutral. However, we demonstrate robustness to this assumption in our simulation study. So as we pointed out earlier, allelic dropout is a common challenge in single-cell DNA sequencing. So we explicitly model this process of the alleles dropping out as the latent VAF. So this latent VAF Yij is dependent on the VAF Xij and the doublet status Zi. And the interesting thing about allelic dropout is that we can actually use it as a further signal to decipher between singlets and doublets. Because when alleles drop out in doublets, the latent VAF Yij is actually going to take on an even greater range of values than when it's a singlet, such as 1/3 in this example here, or 2/3, which we won't see in singlet droplets. So finally, in the last stage, we model the generation of sequencing reads using a beta binomial model that also accounts for the false positives and false negatives reads resulting from the amplification chemistry. So using this generative model, we determined for each droplet I the most likely value of the doublet status Zi via a closed form expression that marginalizes out the value of the VAF X and the latent VAF Y. And so this will provide an answer to our question as to whether the droplet is a singlet or a doublet. So to assess the performance of doubletD, we performed a simulation study, including benchmarking against a single-cell RNA sequencing method called Doublet and a single-cell genotype inference method, SCG, which also has a doublet detection mode. Additionally, we looked at doublet detection in a mixture of cell lines, as well as the impact of doublet removal on phylogeny inference. However, due to time constraints, I won't have time to discuss this last result, but they can be found in our paper. So our simulation study was designed to answer 3 questions. So the first is, how sensitive is doubletD to the parameters of the experimental design? The second is, our method robust to copy number aberrations? And again, due to time constraints, I won't be showing these results today, but we did find robustness to copy number aberrations during our simulation study. And perhaps the most important question, will removing doublets help improve downstream analyses? So we generated 500 in silico droplets with the associated total invariant read count data by using an evolutionary model with 10 clones and varying key parameters such as the number of mutations, the doublet probability, the allelic dropout probability, as well as the sequencing coverage. So we did 5 replications for for each combination of parameters for a total of 405 experiments. So we found that doubletD performed well in all experimental regimes, outperforming both Doublet and SCG doublet in terms of F1 score. In addition, doubletD had the highest recall without sacrificing precision, and this is because of doubletD's ability to actually identify the nested and self-lit doublets, whereas the other methods do not perform as well. In addition, we noticed that Doublet is the worst performing method, indicating that direct application of RNA sequencing methods for doublet detection may not be optimized for DNA sequencing data. So next, we examine if the removal of doublets with doubletD can improve downstream analyses, such as clonal genotype inference. So in particular, we sought to compare the ability of SCG to infer the ground truth genotype set in 3 different ways. So first, we ran it in its singlet mode, which doesn't account for doublets. Then we ran it in the doublet mode, which is a more complex model that also models the presence of doublets. And then finally, we looked at what would happen if we use our method doubletD to identify and remove the doublets, followed by running SCG in singlet mode. So we found it very encouraging that a combination of doubletD plus SCG singlet actually outperformed both modes of SCG and facilitated more accurate genotype inference. And this is because SCG doublet tends to be a bit more conservative and calls fewer genotypes than what was actually present in the ground truth set. Whereas on the other hand, SCG singlet actually created a number of novel genotypes to account for the doublets that were present in the data. So lastly, we evaluated doubletD on a mixture of about 1,500 droplets, comprised of 50% KG1 and 50% Raji cell line. And although we don't have ground truth, we specifically chose this data set because of its unique properties allowed us to orthogonally identify 42 droplets that we suspected are neotypic doublets. And this is because this data set only contains 2 clones that are arising from distinct origins, which was a helpful tool in helping us identify these neotypic doublets. So we again benchmarked our performance against SCG and Doublet. And as you can see, doubletD was able to identify all 42 of these neotypic doublets. SCG doublet also performed well, but did end up missing 2 of these doublets, whereas Doublet was only able to identify 2/3. So in conclusion, we introduced doubletD, which is the first standalone method for doublet detection in single-cell DNA sequencing data. We showed that it outperforms existing options and reduces complexity of downstream methods while improving performance. Lastly, we note that with effective doublet detection post-sequencing, doubletD has the potential to increase sequencing throughput as well as lower reagent costs of single-cell DNA sequencing. In addition, there are a number of promising directions for future work. For example, we are interested in extending doublet detection to ultra-low coverage single-cell DNA sequencing. And lastly, I would like to again acknowledge my co-authors, Palash Rasuto and Professor Mohammed Al-Kabir, as well as our funding, and also thank the members of our lab for their helpful feedback on this work. Thank you so much for your time, and I'll be happy to take any questions that you have.